also cover some aspects of using DNA-based methods to developing an effective early warning system. However, this work is specifically on the larval settlement stage of crown thorn starfish. Most of this work was done by our group of researchers at the ARC Centre of Excellence for Cory Studies at JCU, and the molecular techniques were developed in collaboration with Sven Uthicki and Jason Doyle at Ames. Funding was provided by RRC at the start, and also the Morris Family Foundation, the NESP Hub, and the Ian Potter Foundation through the Lizard Island Research Station. So, as we all know, crown thorn starfish are a major contributor to coral loss on the GBR as a result of their propensity to undergo these major population eruptions. And as a result of this, studying the supply side ecology as well as the drivers of these population eruptions have been a perennial research activity since the 1960s. The exceptional reproductive potential of this starfish suggests that even small changes in key demographic rates could trigger these outbreaks and population eruptions. So in the past, these eruptions have been attributed to either single mass recruitment events or progressive accumulation of starfish over successive years of elevated recruitment. And these sudden versus progressive increases in population size quite likely have very different explanations, but this really highlights the critical importance of assessing these early life history processes that influence the replenishment as well as the dynamics of these populations. One of these key early life history processes that are affecting population replenishment is larval settlement, which is the passage from the planktonic to the benthic habitat. It really starts with a behavioral search for suitable substratum to settle on and ends with the actual attachment onto the substratum and triggering metamorphosis. As in other benthic invertebrates, settlement plays a key role in determining the replenishment and dynamics of these populations. The large spatio-temporal differences in adult densities that we clearly see on GBR reefs are almost certainly linked to differential rates of settlement and recruitment. We just don't know how they are linked yet. To understand the drivers of the high recruitment pulses and ultimately the differences in the adult densities, there's clearly a critical knowledge gap there. However, when we look at how we've been progressing with resolving persistent information gaps, it doesn't really look, doesn't exactly look great. So Morgan already mentioned that study from 2017 earlier, where they went back to Peter Moran's list of key COTS research questions. And I just wanted to quickly highlight that we, in fact, still haven't answered 2/3 of the key research questions that are pertinent to the settlement stage, or at least the very early post-settlement stages of crown thorn starfish. This is particularly striking given from what we've been researching on other echinoderms. We do know of the importance of these life stages to managing, predicting, and understanding these eruptions that we know from other echinoderm species. The problem is COTS settlers are very cryptic and incredibly small, so the difficulty of actually detecting and studying planktonic larvae, settlers, and small juveniles in the field has constrained our ability to actually address these pertinent and important information gaps in the past. So in past studies, what happened quite often is that inferences about settlement and juvenile abundance had to be made on the densities and the distribution of older juveniles or even adult populations. More recently, however, important progress has been made, as already outlined by Sven, with COTS larvae being detected and quantified in plankton samples using molecular techniques, as well as genworms and other people demonstrating that newly settled 6+ month-old juveniles we are actually able to effectively sample them in rubble habitats using fine-scale visual surveys. However, progress has really yet to be made in the field study of COTS settlement. So in this paper, we introduce an innovative method to record COTS settlement using these purpose-built artificial settlement collectors that I'll show you in a minute, and combining these collectors with the methods using the COTS-specific MT-CO1 primers that Sven already outlined, as well as droplet digital PCRs. So the purpose really was firstly to confirm that COTS settlement can be measured using this method, as well as explore some preliminary spatial and temporal patterns on the GBR. So for our larval settlement collectors, we took these aquarium filter bioballs, which have a really high surface area, and basically just sandwiched two of these leaf basket collectors together and filled them with 60 to 70 bioballs per apparatus. And then during deployment, each collector was secured to the substratum using stainless steel stakes and wire rope. And this method really was just a modification from previously used samplers by people like John Keesing on the GBR from Ames and Jose Hernandez in the Mediterranean on the urchins. It's just a bit more robust version of that. And then prior to retrieval of these, after the settlement season, each collector is covered in a fine mesh bag, and then back on the boat or the research station, the mesh bag, as well as the entire leaf collector apparatus, as well as all the Bibles contained therein, are processed in really specific buckets, and then all the biological material that was contained in the apparatus is stored in ethanol until we process it. So developing a suitable DNA extraction protocol proved to be quite difficult because of the large volume of the sample contained per collector. However, after a couple tries, the following extraction method that I'm presenting did result in a successful amplification of the COTS DNA among the sample. So all the collected material was stored in ethanol, and then later we drain it and vacuum freeze-dried and use these NutriBullet high-speed blenders to homogenize the sample, and then we subsample the homogenized sample material and use a modified version of the Wayne's extraction method to extract the DNA from the smaller subsamples. And then we use the extracted DNA as well as the COTS-specific forward and reverse primers that Jason and Sven developed, and ran the droplet digital PCRs at Ames for all the samples. So far, all the modeling was done based on presence of copy numbers above a conservative threshold for each sample, as some more extractions and validations need to be done to report absolute numbers of settlers. So far, a total of 219 collectors were deployed on the GBR over 4 sampling periods from October 2016 to February 2020. The sampling periods coincided with the COTS summer reproduction period, So the collectors were deployed prior and retrieved after the predicted spawning. We have a pretty good understanding of that. All collectors were deployed at sites with relatively steep rubble slopes, which is where we have been predominantly finding those early small-sized juveniles, and which usually have a very high coverage of CCA encrusting, rubble encrusted with CCA. As you can see, we mainly focused on reefs off Townsville and off Cairns in the first couple of years, and then extended our sampling to 3 reefs off Lizard Island in the 2019-2020 summer. Out of the 193 settlement collectors that were successfully retrieved and analysed as part of this study, COTS settlement was recorded in 60 collectors, which equates to about 31% of samples. During the 3 sampling periods from 2016 to 2018, settlement was recorded at all the 3 sampling reefs, which were located in the central and northern GBR. And then during the 2019-2020 sampling period, all the 7 reefs off Townsville recorded a fair bit of settlement. However, only one of the 3 reefs off Lizard Island, the northern GBR, did. We observed considerable interannual variation in settlement at Rib Reef, where we continuously went back throughout all the different sampling periods. Among years, COTS settlement probability was highest between October 2016 and January 2017. And there was a lot less settlement 1 year later and 3 years later during the more recent sampling periods. Throughout these 4 years, the adult densities at Rib Reef were above what we would consider outbreak densities. However, they also did decline throughout these years. So seeing an interannual decline over that time period is not particularly surprising. The extent to which COTS larvae settle in shallow versus deeper coral reef environments has been a quite controversial but really unresolved research question over the last 2 or 3 decades now. We did not find any effect of depth on larval settlement here, with the COTS settlement collectors predicted to be broadly similar among the 3 depth ranges. For logistical reasons, we still haven't deployed them any deeper yet. However, in contrast to the deep water recruitment hypothesis, I think it's quite clear to say that they do readily settle in shallow reef environments overall. Recently published records of 6+ month-old juveniles, small juveniles across a wider depth range really support this conclusion. Although the mean density of juveniles was highest between 8 and 14 meters here. This is some work done by Jennifer Wilms, and this really suggests that it's likely the presence of particular reef substratum and associated chemical cues rather than things like depth that is driving these small-scale settlement patterns of crown thorn starfish on reefs.
Sorry, Peter, you've got 2 to 3 minutes left.
On a larger spatial scale, substantial latitudinal variation in settlement was observed. Our model predicted collectors on reefs located in the central GBR to be more than 10 times as likely to record settlement compared to the northern section near Lizard Island. This latitudinal difference in settlement rates between the northern and central GBR is also clearly reflected in adult densities during this summer in 2019 and 2020. However, it's also important to emphasize that there was some settlement around Lizard Island where COTS densities are clearly very low now in this past summer. I think importantly, this recent progress allows us to address some of these really important but persistent information gaps. There are fairly consistent differences in the incidence of population eruptions among reefs on the GBR, so comparing settlement and settlement substratum availability may actually explain some of the ecological drivers of these eruptions and differences between reefs, and help us establish which reefs are likely or rather unlikely to be exposed to mass recruitment as well as high adult densities. Secondly, this method also provides an opportunity to add settlement as a variable in demographic models and assess the stock recruitment dynamics of this boom and bust species. Notably, exploring the links between larval supplies, settlement, juveniles, as well as adult densities could also help us reaffirm us that just culling adults and not trying to actually address any of the other life stages is the most effective way. We do now know, as Morgan outlined this morning, that large-scale movement is very unlikely. So what this means is that understanding these settlement patterns is likely crucial to our understanding of the eruptive population growth and successful management of crown thorn starfish. So most importantly, this method has application to the early detection of new and renewed population eruptions. To date, eruptions really only become apparent when high adult densities are detected on reefs on the GBR. Annual deployments of larval settlement collectors are on across a hierarchy of GBR reefs could detect localized fluctuations and sharp increases in settlement and recruitment, and therefore herald new as well as renewed population eruptions. This could really prove to be the difference between having to manage the manageable task of controlling small COTS densities on a handful of reefs in one region compared to the rather tricky task of population control across an entire region or multiple regions with high adult densities on pretty much all the reefs. So in conjunction with annual monitoring of planktonic larvae as well as juveniles and or other monitoring tools, I think we definitely have a chance here to address this and develop an effective early warning system for future COTS population eruptions. Yeah, just quickly thank you to all the funding bodies as well as the institutions and the people involved in this research.